Introduce our last but not least speaker of this semester, Dr. Natthida Petchampai, a postdoctoral fellow in our department. Natthida was born and raised in Thailand. There she received her bachelor and master degree in biochemistry. She worked as a research assistant and medical technician before moving to SA. From 2006 to 2008, she worked as a research assistant in Dr. Macaluso labs at Louisiana State University. After 2 years of working in his lab, he was admitted into the PhD program at LSU, where she earned her PhD in the spring 2013. Her scientific training included research with different biological systems, such as virus, bacteria, and tick cells. Natthida attended and presented her work at national, local, international scientific meetings and published a total of 13 articles in peer-reviewed journals. Natthida is involved in several projects in my lab. However, today she will discuss the allosteric regulation of tyrosine Egypti pyruvate kinase 1. She will also highlight the key role that this enzyme plays in both carbon and nitrogen metabolism in mosquitoes, and this is part of ongoing collaboration with Dr. Pizarro lab.
Good afternoon, everyone. Thanks for coming. So today I will present part of my research project, allosteric regulation of Aedes aegypti pyruvate kinase 1 and its implications in carbon and nitrogen metabolism. So here is an outline of my talk. First, I will give you some background of Aedes aegypti mosquitoes, its ammonia metabolism, and pyruvate kinase or PK enzyme, which is the enzyme that I'm studying. And then I will present you some of my results, including PK protein expression, purification, crystallization, 3D structure determination, and PK kinetic studies. And at the end of my talk, I will give you a summary, conclusion, and future directions of my talk. A little bit about Aedes aegypti. So Aedes aegypti is the mosquito that is most frequently found in tropical and subtropical areas of the world. In the US, it has been found in a number of states, as shown in blue in this map created by CDC. This species of mosquito is considered to be a primary vector of several viral diseases, such as dengue and yellow fever, Zika, chikungunya, and because they have adaptations to the environment that make them highly resilient, it is very difficult to control or eliminate this species of mosquito. Therefore, better understanding of mosquito metabolism is important for more effective vector control. So in our lab, we study ammonia metabolism in Aedes aegypti. As you all may know, that female mosquitoes must feed at least one blood meal in order to produce eggs. However, a majority of the amino acids derived from blood meal proteins are oxidized for energy production, as shown in this picture. And an oxidation of the amino acid oxidation, however, created a large amount of ammonia as a byproduct. But mosquitoes don't have urea cycle like other animals do, and the high level of ammonia is toxic to the cells. So this leads to the questions of how these mosquitoes detoxify ammonia. Previous studies from our lab have shown that female mosquitoes remove ammonia through the synthesis of amino acid and excretion of uric acid, allantoin, allantoic acid, and urea. This diagram shows the ammonia metabolism in Aedes aegypti mosquitoes. You can see that female mosquitoes use multiple metabolic pathways to detoxify ammonia, and several enzymes, including glutamine synthetase, glutamate synthase, xanthine dehydrogenase, and alanine aminotransferase have been shown to be involved in ammonia metabolism in AD egyptides. Previously, we found that knockdown of this enzyme, alanine aminotransferase, resulted in an accumulation of uric acid in mosquito midgut, and also result in a delay in blood digestion, nitrogen waste excretion, and oviposition with a decrease in egg production. So what is alanine aminotransferase? This enzyme, you can see here that it catalyzes the conversion of alanine and alpha ketoglutarate to pyruvate and glutamate. This reaction is reversible. That means the enzyme can catalyze the conversion of pyruvate and glutamate to alanine and alpha ketoglutarate as well. And this pyruvate can also be obtained from like glycolytic pathway, as you can see in this figure. This is the pyruvate in glycolysis that I just talked about. You can see that this pyruvate can be used as a substrate of alanine aminotransferase enzyme in ammonia metabolism. And in glycolysis, this pyruvate is produced by the enzyme pyruvate kinase, which is catalyzed the conversion of phosphoenolpyruvate and ADP to pyruvate and ATP. And the product pyruvate of pyruvate kinase here is very important in cellular metabolism because it can feed into a number of metabolic pathways such as ammonia metabolism in mosquitoes. In addition, recently we found that glucose oxidation through glycolysis is important for ammonia metabolism in AD egypti. In this study, we fed female mosquito with blood meal supplemented with stable isotope level glucose, and then we measure the downstream metabolite here. And we found that glucose was used for ammonia detoxification and uric acid synthesis through multiple metabolic pathways here, including glycolysis. And pyruvate kinase is the key enzyme in glycolysis here. So, and that's why the aim of my study is to characterize this enzyme. So what do we know about pyruvate kinase? Pyruvate kinase is a key regulatory enzyme in glycolysis. So it has been studied extensively in mammals and several organisms. For PK activity, we know that the activity of the enzyme is controlled in most organisms by one or more allosteric effectors. And the major allosteric activator of this enzyme in most organisms is fructose 1,6-bisphosphate. However, other sugars like fructose 2,6-bisphosphate, ribose 5-phosphate have been shown to regulate the activity of this enzyme. In insects, the kinetic parameters of PK have been determined, but there are currently no structural data for insect PK. In mosquito, we know that PK activity has been detected during embryogenesis, but there is no report of PK activity in adult mosquitoes. For PK genes, almost all organisms have at least one PK gene. In human, 4 PK isoforms are expressed by 2 genes, which are PKLR and PKM. In AD aegypti mosquito, we found that there are 2 alternatively spliced mRNA variants that code for PK enzyme, and these are AAPK1 and AAPK2. This is the sequence alignment of the AAPK1 and AAPK2 isoform. You can see that they have only like 16 amino acids different between the 2 isoforms of pyruvate kinase in AD aegypti. And for my study, I chose to characterize the AAPK1 isoform. So now let me show you some of my results first we express PK protein and purify, and these are the summarize of the method that I use. I first clone the AAPK1 gene in the expression vector and transform it into Rosetta 2 E. coli. The E. coli cell were then grown at 37 degrees Celsius, and the protein expression was induced with IPTG at 25 degrees Celsius overnight. And then we collect the E. coli cell pellet and resuspended in the lysate buffer to lyse the cell. And the lysate was finally centrifuged and purified by nickel affinity and side exclusion chromatography. And this is the result of side exclusion chromatography. The chromatogram on the left is the elution profile of the purified recombinant AAPK1. The fractions within these peaks were the purified enzyme, and these fractions were collected and tested for enzyme activity, and we found that our enzyme is active. And the picture on the right is the chromacy staining of the protein before and after purification. Lane L is the proteins before purification, and lane P is the protein after purification. You can see here is the purified enzyme with the molecular weight like above 50. We expected the molecular weight to be about like 57 kilodalton. So after we got the recombinant purified enzyme, we then try to crystallize the protein and determine its 3D structure. So we use the sitting drop vapor diffusion method to crystallize the AAPK1 enzyme. And for initial crystallization, we screen for the crystallization reagents that can give us like a good crystal. This is just an example of the crystallization reagents that we use. Out of like 96 conditions, only 6 conditions that I highlight in red here gave us the crystal, but like these crystals are not even all of them like give like a good X-ray diffraction data. So we then have to optimize the condition of this crystallization reagents by varying the salt concentration and the precipitant reagent. And this is just some pictures of the crystal that we got. And after we got this crystal, we then flash cool in liquid nitrogen, cryoprotected in crystallization reagent, and then submitted for diffraction data collection. And this is the picture of the AAPK1 crystal in a cryo loop. And here is the picture of pyruvate kinase 3D structure. The picture on the left is the AAPK1 monomer. You can see that there are 3 domains, which are domain A, B, and C. And this is the fructose 1,6, the sugar fructose 1,6 bisphosphate that we can see like bind to the monomer of the AAPK1. But this enzyme is a tetrameric protein, and the figure in the middle here show the tetramer organization of AAPK1. See like 1, 2, 3, 4 here. And the picture on the right is the overlay of AAPK1 with the 3 human isoform PKM1, M2, and R. And this is the alignment of the PK sequence from AD aegypti and human PKs. So the secondary element on the top represent the secondary element from like AAPK1, and from the bottom are those from PKM1, like human PKM1. enzyme. So these two, the AAPK1 seem to be like more similar to the human PKM1 isoform. As you can see in this table, this is the sequence and structure similarity between AAPK1 and human PK isoform. So like in this table, the sequence identity similarity percentage are shown below the diagonal here for all enzyme, and the structure similarity is shown above the diagonal here. You can see that the AAPK1 is more similar to the non-allosteric human like PKM1 isoform, and this finding raises to the question if our enzyme is not allosteric regulated, because like PKM1 is not allosteric regulated, or it might be like it might be that the enzyme is regulated by the other special effectors. So then, like in order to test if like our enzyme is regulated by the usual effectors or a unique effector, we perform PK kinetic study. And so in order to run the enzyme assay, we use like this technique, which is the lactate dehydrogenase coupled assay system. You can see here that pyruvate kinase can catalyze the conversion of ADP and phosphoenol pyruvate to ATP and pyruvate. And in the presence of like NADH, the pyruvate is converted to lactate by enzyme lactate dehydrogenase, and we measure the decrease in the absorbance at 340 nanometer resulting from oxidation of NADH. And this is the result. So like we test the effect of several sugars on AAPK1 kinetic parameter. So these are the list of the sugars that we test. So we pick like, this major allosteric effector in other organisms, and we also pick the 5 carbon sugar and 6 carbon sugars. And and these are the kinetic parameters that we determine. So the K prime is calculated as K half to the H power. And K half is the concentration of the substrate that produce a half maximal enzyme velocity. And Hill coefficient here is the Hill slope. So when Hill coefficient or H is equal to 1, this equation is the same as the Michaelis-Menten equation. And when it is greater than 1, the curve is sigmoidal due to positive cooperativity. So in this assay, so I prepare pyruvate kinase activity assay, and so with no effector, you can see that the K prime is about 0.25 and the Hill coefficient is about like 1.08. And once I add that this sugar fructose 1-phosphate, the K prime decreased to 0.06. This means that this effector activates the enzyme, and you can see that the Hill coefficient is increased. And the other sugar that affect AAPK1 activity is ribulose 5-phosphate, but it affects in the opposite way. You can see that the K prime is decreased to 1.13 compared to no effector. So we can say that this effector inhibits the enzyme. Next, we test the effect of the amino acid on AAPK1 kinetic parameters, and several of amino acids have been tested, and most of these amino acids have been shown to be important in nitrogen metabolism in mosquitoes. And we found that alanine, glutamine, proline, and serine decrease the K prime, and the Hugh coefficient is increased here. So we conclude that alanine, glutamine, proline, and serine show a classic allosteric activation with a decrease in the K prime and no effect on the Vmax. Sorry, I don't have the Vmax here in this table. So because pyruvate kinase, because both sugar and amino acid can bind to pyruvate kinase to inhibit or activate the enzyme. So our next study, we try to see if the combination of the amino acid and the sugar affect the enzyme activity in a different way. So the amino acid, the first amino acid that we test is alanine, and we test the combination of alanine with ribulose 5-phosphate or fructose 1,6-bisphosphate or glucose 6-phosphate. So you can see that alanine here, if you add only alanine, the K prime is decreased. But when you add only ribulose 5-phosphate, the K prime is increased. So this means alanine is the activator, but ribulose 5-phosphate is an inhibitor. But when you add alanine and ribulose 5-phosphate together, you can see that the K prime is about the same as is shown in alanine. So this means that alanine prevents the inhibitory effect of ribulose 5-phosphate on the enzyme activity. And when you add fructose 1,6-bisphosphate and glucose 6-phosphate, I mean, this is a different reaction, but it gave a similar result. So you can see that the K prime is increased when you compare to alanine alone. So I conclude that F16PP and G6P diminish the effect of alanine on AAPK1 activity. The next amino acid that I tested is glutamine. So glutamine alone show the same effect with alanine, which is the decrease the K prime of the enzyme. But when you mix glutamine and ribulose 5-phosphate together, you can see that the K prime is increased compared to glutamine alone. So, but when you compare with ribulose 5-phosphate, you can see that the K prime is decreased compared to ribulose 5-phosphate. So I conclude that glutamine prevents the inhibitory effect of ribulose 5-phosphate on the enzyme activity. And for fructose 1,6-bisphosphate, you can see that when you add fructose 1,6-bisphosphate in the presence of glutamine, the K prime is increased when compared to glutamine alone. So this tells me that fructose 1,6-bisphosphate diminishes the effect of glutamine on the enzyme activity. The next amino acid that I tested is proline. So these amino acids that we talk about are important in nitrogen metabolism in ADH5, and that's why we pick this amino acid. So you can see that for proline alone, the K prime is 0.02, and ribulose 5-phosphate alone, the K prime is about 1. But when you mix them together, you can see that the K prime is decreased compared to R5P alone. So this, I conclude that proline prevent the inhibitory effect of R5P on AAPK1 activity. And another one is fructose 1,6-bisphosphate. So when you add fructose 1,6-bisphosphate in the presence of proline, you can see that the K prime is increased compared to proline alone. And I conclude this that fructose 1,6-bisphosphate diminish the effect of proline on AAPK1 activity. And the last amino acid that I test is serine. This amino acid serine alone decrease the K prime compared to no effector. But once you you mix serine and ribulose 5-phosphate together, you can see that the K prime is decreased compared to ribulose 5-phosphate alone. So this tells us that the serine prevent inhibitory effect of R5P on the enzyme activity. And the result of the combination of serine and fructose 1,6-bisphosphate and serine and glucose 6-phosphate are very similar in that the K prime is increased when compared to serine alone. And this tells us that fructose 1,6-bisphosphate and glucose 6-phosphate lessen the effect of serine on AAPK1 activity. And these are the summary of all of the results that I just talked about. You can see that fructose 1-phosphate is an activator because it increases the affinity to the substrate of the enzyme. Ribulose 5-phosphate is an inhibitor. These 4 amino acids are an activator as well because it increases the affinity to the substrate. But when you mix each of these amino acids with ribulose 5-phosphate, so this amino acid prevent the inhibitory effect of this sugar on the enzyme activity. And this sugar, fructose 1,6-bisphosphate and glucose 6-phosphate, if you add it in the presence of these amino acids, you can see that the affinity of the enzyme to the substrate is decreased. This means that this sugar lessen the effect of this amino acid on the enzyme activity. Okay, I just talk about the summary. But so in summary, the 3D structure of the recombinant AAPK1 was determined by X-ray crystallography in complex with fructose 1,6-bisphosphate. And we found that AAPK1 is allosteric controlled by multiple effectors. And I just mentioned fructose 1-phosphate is an activator, but ribulose 5-phosphate is an inhibitor. These amino acids are an activator as well. And these amino acids prevent the inhibitory effect of ribulose 5-phosphate on enzyme activity. And these two sugar, fructose 1,6-bisphosphate and glucose 6-phosphate, alter the effect of the amino acid on the enzyme. So in conclusion, our study show how specific amino acids and phosphorylated sugars tightly regulated the activity, the catalytic change of AAPK1, therefore uncovering a unique regulatory properties of the enzyme. And since nectar and host animal availability in nature may be unpredictable, the distinctive AAPK1 allosteric regulation could enable metabolic control of carbon and nitrogen flux in sugar and blood-fed mosquito, and therefore it could represent a critical regulatory point within mosquito metabolism. And the unique AAPK1 allosteric properties support a complex role for this enzyme in the mosquito metabolism. So I would like to thank Dr. Scarfield for her help and support throughout the project, and also students in Scarfield Lab. And I'd like to thank Dr. Pizarro for the help with the X-ray crystallography, and Calibel for the help with protein. Dr. Izzo at University of Arizona for the help with RNA stuff. And would like to thank Dr. Dagan and at Israel Institute for Biological Research, Dr. Horvath, Dr. Lorenzi, and Dr. Hawk at the University of Texas MD Anderson Cancer Center for the help with mass spectrometry. And this research is financially supported by Corinne Adams Baines Professorship, Carol Levin Bernick Faculty Grants for Fellowship, Vector-borne Infectious Disease Research Center Pilot Grant, and the NIH Grant. And thank you all for your attention, and I'll be happy to take any questions. Yes.
The AAP2. Also, why does the mosquito have those two forms that are so similar?
Yeah, and like I don't know exactly. We plan to work on like this other isoform of the AAP of the pyruvate kinase enzyme as well. Like the difference of the amino acid is only like 16 of the protein is only like 16 amino acids. So it's possible that it will be similar, but who knows, like only 16 amino acids could be like critical for the structure, you know, of the enzyme. So it might be different, like we don't know. Yes.
Maybe you mentioned it and I missed it, but how similar or different are those genetic properties compared to the human protein?
Oh, I didn't mention actually. I don't remember exactly the the number. But I think it's different, but I don't remember like, you know, like how much different.
And also they have not studied the combination of both sugar and amino acid. Unique. Nobody has reported that, which for mosquitoes might be very important because there are both sugar feeders as well, blood feeders, especially the females. So those studies have not been done. But for the other ones, you know, it's very
like so the kinetic properties here in mosquito is very, very unique for the mosquito. Like alanine for mosquito is an activator, but in human it's an inhibitor. And but like serine is an activator for the mosquito, but in human it's an activator as well. Yes.
The different effects, especially when you hear them for the first time, are confusing and hard to understand. Can you help us think about this in terms of how the pieces fit together? Presumably, there's been selection over time for these different interactions. What's going on and what's driving this? What are the selective forces, or is it just too early to know?
So, like you said, like what drive the difference between the between the mosquitoes and, like, let's say human or whatever. I don't know exactly why, but like so think about like so mosquitoes like can be both like can feed on both sugar and blood fat. So like and in nature, like, you know, like the source of the food is not always available, so it's unpredictable. So like in mosquitoes, they might like adapt into the point that they have to survive. Like, for example, if they cannot find sugar, they're gonna have to do something. For example, decrease the activity of the enzyme, and when they found blood it might be different. So I think like this food source and like the environment probably like influence like how the enzyme is like regulated in mosquito and different in from the other organisms.
So just really maybe things to easier think about it. So in humans there are 4 isoforms, but only one of the isoform is allosterically regulated, PKM2. So that the PKM2 is the form that you only find in fetal tissue. Normally adults, all of us, we only have PKM1, which is a non-allosterically regulated enzyme. So the way to think about it is that when you have a cell that is developing and is growing very fast, so what you want to see is you want to have something that gives you metabolic control without having to change the gene expression pattern. So you have some enzymes that will have some kind of built-in regulatory mechanisms, so you can adapt to the conditions, to the different the presence of different substrate and products. So that way you can have a regulatory mechanism within the cell that doesn't have to depend on gene expression of a new genes. So what happened is, for example, in cancer cells, the cancer cells shut down the PKM2 expression and shut down the PKM1, and then they recover this PKM2 expression of this enzyme. And the people that have been studying cancer, they think this is advantageous for a cancer cell because it allows the cancer cells to, when there's a lot of oxygen present around, and they can go to the glycolysis and they can produce a lot of ATP, but then because cancer cells kind of grow, tumors grow irregularly, so there's sometimes there will be not enough oxygen available, so then you cannot go to glycolysis because you can create too much, you don't have enough oxygen around. So what you can do actually is, just kind of shut down the glycolysis pathway if you have, without having to change, because you have an allosterically regulated enzyme that will depend on this fructose biphosphate, so they can diverge. So it's kind of this balance between catabolism and anabolism that will kind of allow them to do without having to kind of changing the gene expression pattern. That is the idea, that what you have these things kind of associated with enzyme kinetics. That is a way that you can think about it. It's kind of like a cell will kind of like optimize its metabolism without having to kind of depend on expression of new genes or expression of other regulatory products. That will take a lot of time, and then it can allow them to kind of be adaptive in that way. So you can think about it that kind of like applies for the mosquito.
But it's true, whatever you say, it's too early to see how exactly it's regulated. The answer is yes. That's why, right now, Natthida, well, not right now, it's just has done a lot of work on PK activity in tissues, different tissues. So we wanted to know how the enzymes work in different tissues and under different diets, including starvation, longer starvation.
And the Krebs cycle is working all the time in this organism. It doesn't shut down at all.
I would say yes, but I think depending on the condition, because if they are under extreme starvation, the cells might need to think about how to do, right? Same thing for the
enophilins do that to some extent.
And also the post-translation modification. We are interested.
Yes.
So if in humans, the different isoforms are active at different life stages, where I think you're saying 2 is fetal tissue and then the other ones are in adults, do you think there's something similar going on with the 2 isoforms in the mosquitoes where they're expressed at different life cycle stages depending on growth and everything?
It could be. It could be because like when we clone this enzyme, we use like we use like larva pupae, like adult. So like we cannot say this is like specific for only adult. But it could be that like in larva is different, but we haven't tested yet. I mean, that's why they have 2 isoforms. Gotta be something.
question? Thank you so much.
Thank you.
We will see you again next fall.